Oxygen, 3% is carbon dioxide and other gases. And I'll put everything together. So, nitrogen, a huge amount sitting up in the atmosphere, your plants can't access it. So, we have to go through the process of fixing that nitrogen. Be really, really careful with the terminology here, because so many people mix up this term, nitrogen fixing, with nitrifying, the process of nitrification. Completely different process as nitrogen fixing. So isn't it fun? Try to keep those two things separate. When we're dealing with taking into gas and converting it into a form that your plant can use, we have both symbiotic nitrogen fixation, where we're going to be looking at rhizobium in combination with the legumes that it can grow on. We also want to be looking at free-living nitrogen fixation. There's a lot of similarity between the two processes, but there's some significant differences as well. You probably should know what those two differences are. When we're looking at rhizobium, your roots growing down into the soil, on the root hair is where the lecithins are. So your plant has to make a specific lecithin that the rhizobium can bind with. So legumes are the only thing that have these specific lecithins on the root hairs. They're only on the root hairs. So here's our little bacterium. There's our rhizobium. It has to lock and key, so it's like an antibody-antigen interaction. And once that identification has occurred, then this rhizobium can now start to grow through the cell membrane, the cell wall, and this infection thread. So each one of these little round needles are the individual bacteria. So we've got a rhizobium, another one, and it multiplies, and it multiplies. And so it's slowly growing towards the main root. All of the foods for this growth is coming from your plant. So sugar is supplied from photosynthesis up above ground. When that infection thread, so infection thread sounds bad, doesn't it? Well, it's the same process that any pathogen follows. It's just that in this particular situation, this interaction has turned into something beneficial for both sets of organisms. So the infection thread, when it reaches your main root, these rhizobium put out hormones that cause massively more sugar. So should I do sugar? Now comes down into that root hair. Well, if we have just literally boatloads of sugar arriving in that little root hair, those bacteria just start going ballistic. They start growing till there's billions and billions, and they cause that root hair to swell up. So we end up with a great big nodule. So there's the nodule that we should be seeing on any of our nitrogen-fixing legumes. Billions and billions of bacteria are growing inside that nodule. And of course, when you get bacteria growing really, really rapidly on sugar like this, what happens to the middle of the nodule? It's going to go anaerobic. And now, when this is anaerobic, so the middle of the nodule is now anaerobic, that allows these bacteria to express the nitrogen fixation, and we abbreviate that in the world of science as NIF, nitrogen fixation genes. So in the middle of that nodule, whatever nitrogen is moving in there is now going to be ripped apart. The oxygen's all used up out here because that process of nitrogen fixation can only occur in anaerobic conditions. So that NIF gene starts to work. It's going to take that nitrogen that is triple bonded to another nitrogen. And it takes a lot of energy, some very special enzymes, to supply the energy to rip those two guys apart. So now we have a nitrogen plus another nitrogen. What's your plant going to do with that? Or what's your bacterium going to do with that? It's in the middle of the nodule, so it's the bacteria that are going to do this. What does the bacterium need in order to grow? Nitrogen. Yeah, and what is nitrogen with a carbon? So here's our sugar, and so it's coming from our plant, and your bacterium is now going to fix that nitrogen onto that carbon chain. It's going to have to pull in two hydrogens in order to do that. You know, and so on another carbon, maybe in the same chain or a different one, we're going to bond that other nitrogen, and we got to pull in two hydrogens. So what's happening to the pH within that nodule? It's going alkaline. Typically, nitrogen fixation is a process that occurs in alkaline soils. So it's a strictly bacterial process. The protein, or probably more accurately, the amino acid that is produced, who gets that? We're in the middle of the nodule. Who gets that amino acid? It's the bacterium. The bacteria continues to grow. It's a happy camper. It's having a wonderful time. Only if the rhizobium in that nodule produces excess amino acid does it say, "Oh, plant, you've been so nice. You've been so good to me, contributing all this sugar. Here, have some protein." So now some of these amino acids, some of these proteins get sent back to the plant. When your plant gets that protein, what does your plant do with it? Uses it to grow. So now your plant, putting on extra leaves, putting on extra blossoms, putting on extra growth, and the plant's growing better, the bacterium's growing really well. Life is wonderful for both plant and bacterium. Is any of that nitrogen escaping out into the soil? No way, Jose. Your plant has spent a lot of energy helping those rhizobium out. The rhizobium's getting all that extra protein initially. Your plant finally gets some proteins, some amino acids, proteins. They're not releasing that out into the soil. Both organisms are growing better. Isn't this good? When you look inside this nodule, you can tell when nitrogen fixation is happening in the middle of that nodule. Because if you cut this open, and you look at that open surface of the nodule that you cut, in the middle of that nodule, anywhere that nitrogen is actually being fixed, you see a red color. Nitrogen fixation, those enzymes, have to have a heat molecule. So just like your blood, the inside of that nodule should be bright red, just like a drop of blood. If you're getting excellent nitrogen fixation and you're really improving the protein content of your plant, the whole inside here will be a bright red, just like a drop of blood. Well, what if you cut open that nodule and all you see is a bunch of little pinky veins? Yeah, that's not really doing very well. I'm not impressed with that rhizobium. I want to go find a different species of rhizobium, something that's more indigenous to this place, because I want to get real good nitrogen fixation. But at least you're getting some, so it's better than nothing. What if you cut open your nodule, and the inside of that nodule is green, or white, or black, or brown? you are not getting nitrogen fixation. Think of all the photosynthate that was used by those bacteria, and the plant is going to get nothing back from that expenditure. So are you really getting nitrogen fixation? Well, first of all, you want to see nodules. And I always have to laugh when somebody has pictures of their nodules, and their nodules look like now there's my root system, and their nodules look like this. This is not big enough to be concerned about at all. Here's my clover plant, and those are my nodules. Don't even imagine for a second that you've got any nitrogen fixation going on here. When I'm looking at a root system like this, I want to see my nodules marble size. I want to see nice, big, fat nodules. Now it's worthwhile to cut that open and see if you've got some color inside of that, see if you've got red. So many parts of the Great Plains of the United States, as I wander through, I'm often stopping at the side of the road. My husband thinks I'm crazy, so do my kids. Get the trowel out, dig up some of that soybean, and look at the root systems. There's absolutely no nodules at all. Do not tell me that you're going to get good yields from that soybean. There is no nitrogen fixation going on. Dig up your clover. You get any benefit from your clover? No nodules. You really probably want to go find some rhizobium, put the rhizobium into your compost tea, brew them up, and get them inoculated into that area where you've got legumes. Get the rhizobium growing on the system, or why are you wasting time growing legumes?
How is it that when we use those nitrogen legume, nitrogen fixing ground covers, you're saying that we're getting too much nitrogen into the tree that it's under, and that it's not releasing any out?
So how do you get the nitrogen that's in the plant out into the soil? What's that process? So we're going to have to go through that. We'll keep going through that cycle.
Oh man, I forgot what I was going to say because wait, don't you prune the plant and then it drops the roots slough off a little bit and then makes it a bit?
How many of you prune your clover? How many prune your soybeans?
Yeah.
How many prune your alfalfa? Probably not. So is that the best way to do it? Well, question here first.
Can you add rhizobium to the extract?
You can add rhizobium to the extract, but when you do that, probably what you want to do is soak the seed in the extract, because then the rhizobium adheres to the seed surface. If the rhizobium is not really active and growing, it doesn't tend to get into the soil and colonize the root system. Now that's not based on a lot of data, so that could be a little iffy. So I'd love to have you guys all do those observations. Get out there and try this.
Okay. I remember my question. How can we find some rhizobium? I know that they sell inoculants at the store, but is there a way that we can culture it naturally from around it?
The rhizobium in the nursery, did that come from Hawaii?
No.
No. So where is that originally from? New York State. Israel. North Carolina. Is that an indigenous organism for here in Hawaii?
No.
Let's see if we can find some of our own. So let's go find some native legumes and find the nodules on those root systems. So if you've got that nodule, we've got billions and billions of bacteria inside that nodule. So just take your nodule, kind of crush it up, maybe dry it down a little bit, crush it up, put that into a little bit of water, soak your seed in that water, and get it out there and get it growing. Because now we've got indigenous species that are much more likely to actually fix nitrogen under these conditions with your soils. And these are European plants that we're trying to grow. Alfalfa, the clover, okay, so we got a little work to figure out what's going to actually work best. So, your plant is now elevated in protein. Some part of the plant has to die. So, yeah, if we go out and prune it, like alfalfa, often what I'll do is go and cut my alfalfa and take and then the alfalfa regrows. Well, the rhizobium is perfectly happy working away, adding more protein into my plant. Part of the root system could die, part of the above ground part of the plant dies, and now the decomposers decompose that plant material. So now where's your nitrogen? Now where's your elevated nitrogen? In the bacteria and fungi. It's not out in the soil yet. Those bacteria and fungi, how are you going to get the nitrogen out of the bacteria and fungi and into the soil in a plant available form?
Heat.
They're in our bacteria and fungi in the soil. How are we going to get that nitrogen out of those organisms and into a plant available form?
It's eaten by the predators and they poop it out.
Right there, just follow the arrows. The predators have to eat them, and that releases nitrogen as NH4. If we're in a fungal dominated system, the pH is going to stay on the acidic side, and that means NH4 stays as NH4, as long as it's fungal. Because if we've got lots of extra hydrogen, this nitrogen is going to stay in the form of ammonium. Ammonium is going to be taken up by a perennial plant, if it's close enough to the root system, or if it's away from your root system, hopefully it gets taken up by one of these decomposer bacteria and fungi. And then some predator has to eat it, gets released again, now it gets into your plant, some part of your plant has to die. Decomposers have to decompose that plant material. Protozoa, nematodes, microarthropods release it, here we are at NH4, and round our nitrogen goes, and it's never lost from the system as long as we stay aerobic, as long as we do not kill any of those organisms. Not ever going to be lost. Now, how do we turn that NH4 into nitrate? Different process has to happen. We've got to have nitrifying bacteria. And the only time these nitrifying bacteria make the enzymes to take ammonium and convert it first into NO2 and then into NO3 is if your soil is alkaline. The pH of your soil must be above 7 for these nitrifying bacteria to make the enzymes that will rip the hydrogens off that nitrogen molecule. These nitrifying bacteria don't take up NH4. They just want hydrogen. Pull those off. Get rid of this nitrogen stuff. We've already got plenty. So what these nitrifying bacteria do is stick 2 oxygens on it, and then the next group of nitrifying bacteria stick another oxygen on, and now we got nitrate. These nitrifying bacteria, 2 very different genera, typically are what we're looking at. So NH4, rip the hydrogens off, and that goes to the bacterium to make energy. So nitrosomonas, I don't think you really have to know this, but I'm a microbiologist, and so I gotta give you the name. Nitrosomonas wants those hydrogens. And then it's going to get rid of a waste product. So it's going to take that nitrogen and put 2 oxygens on it. So we have NO2. And then the next nitrifying, the next step, is Nitrobacter. Several thousand species of Nitrobacter. It depends on the climate that you're in, which one you've actually got functioning. And so all Nitrobacter does is get a little more energy out of this process by adding another oxygen and then producing NO3. The enzymes to do this, only are expressed in alkaline conditions, only in soil that is bacterial dominated. Because if it's fungal dominated, you're going to keep NH4. Bacterial dominated, then we're going to be converting at least some of this to nitrate. Great, now we got nitrate. Who uses nitrate? I'll follow the arrows. Our plant, our annual plants, our weedy things, our veggies. the balance of nitrate and ammonia. How much of your root system is still fungal dominated? How much is bacterial dominated? So is your plant going to get the balance that it needs? And then, of course, the plant has to die, decomposed by bacteria and fungi, eaten by protozoa, nematodes, microarthropods, NH4. If it's in an alkaline soil, boom, there we are. Maybe your bacteria and fungi take it up directly if your plant didn't grab it. So round and round and round we go in an alkaline soil. Not that difficult, but everything requires biology. If you kill your organisms off, you're out of luck. You're going to have to put in organic fertilizers on.
Does the process from NH4 to NO2 and 3 take you towards acidic?
No, this is going to take you towards, this process is going to take you towards alkaline. Because these hydrogens are used by the bacterium to make ATP. And those hydrogens are required to produce energy for that bacterium. So there's no release of hydrogen into the soil with that process.
Now that, what you just talked about, going from NH4 to NO2 and 3, is that commonly known in the world of academia that you're going alkaline in that process?
There are a lot of people that are fairly confused about it because they're still growing plants in anaerobic soils. So now they're seeing soil gets acidic. Well, the plant's not healthy, it's not happy, but because they're seeing nitrification occurring there, linking to things that are, it's just a correlation, it's not a cause effect. So they've got to start looking at oxygen concentration in their soil and how healthy their plant is. Now, let's close the last step in the nitrogen cycle. How do we get into gas back into the system? Anytime we go anaerobic, NH4 is going to be converted into NO2 or NH3. Or if we're anaerobic with these compounds, we're going to be producing N2O or NH3. So anaerobic conditions will denitrify. It will take our ammonium and ammonify. So there it goes as ammonia or N2O. Once these gases are released up into the atmosphere, ozone and sunlight converts it into N2 gas. Completed the cycle. The only part of this cycle that is strictly chemical, if you will, is this last step. So go home tonight and go through the nitrogen cycle and see if you can remember every step along the way. It's a little bit complex.
Were you going to add anything about the free living?
Yep, we're going to get to that next. So now, if instead of rhizobium, now we're looking at Actinobacter or Azospirillum or Azomonas or the Frankia, the free-living nitrogen-fixing bacterial species. And there's a whole group of Actinobacteria that have always been included as these free-living nitrogen fixers. The main one of which has been called Frankia. Well, just to make life fun, the whole world of microbiological taxonomy has just changed. All of these bacteria have been moved into completely different families, genus, classes, orders. They're all over the place, someplace else. And I cannot keep it straight what they're called anymore. I'll figure that out someday. But just to let you know, it's not this anymore. It's not Actino. It now has its own order and class order and family. So they aren't Actinobacteria. They are bacteria that grow in filamentous. They grow in a strand like that, but they're no longer in the group Actinobacteria. Okay, just a side note, as a microbiologist, I have to add that. So free-living nitrogen fixers. We still need the root. Amazing. Still need the root. But instead of any clear association between that plant and these bacteria, isn't one. So your root's grown through the soil, is putting out exudates, if, and only if, this bacterium can somehow get hold of a massive amount of sugar. No other bacteria around or something's going on, so that free-living nitrogen fixer gets a whole bunch of sugar. So one bacteria can become 2, 2 can become 4, or 16, and here we go, growing really happily. So as that bacterium's got enough sugar, what happens to the middle of that colony? It goes anaerobic. And now only under anaerobic conditions are the nitrogen fixation genes expressed. So any N2 gas moving into the center of that colony, it's anaerobic. With that, those bacteria can now fix that nitrogen into protein. Sound familiar? Only there's no giving anything back to the plant. There is no bridge for these free-living nitrogen fixers to give anything back to your plant directly. So now how do we get the nitrogen that's been fixed in there back into a plant-available form?
Through the nitrogen cycle.
Yeah, basically, we're just going to jump straight from here to there. So these two bacteria have to be eaten by predators,
Yeah.
and that produces NH4. And if we're in a fungal-dominated system, boom, around and around it comes. If we're a bacterial-dominated system, boom, boom, around and around we go. So, slightly different, a bit simpler. Don't really have to worry about the nodule and do we have, can we cut that bacterial colony open? Because good luck finding that bacterial colony. But that's the difference between free livers and the symbiotic forms. How many plants can be feeding free-living nitrogen fixers? Every single plant on this planet can have this association in their root system. So if we're inoculating these nitrogen-fixing bacteria into our lawns, into our agricultural systems, into any place you're trying to grow a plant, would you ever have to use a nitrogen fertilizer? No. So what is wrong with us that we're not putting these bacteria out into every ecosystem that we can figure it out? Okay, who's making money on this? So let's go isolate some of these things. So there are inocula of these free-living nitrogen fixers available on the market. What we're not real clear on yet is how effective are these nitrogen fixers at actually doing anything to increase nitrogen content in your soil. Data's not in yet. There's some pretty interesting papers that have been written by Josh and David Schimmel, where they've been looking at those kinds of things. So you might look up those two guys. Josh is at the University of California, Santa Barbara, and Dave is at NASA, I believe. Last time I looked. So if anybody's doing that research, it's going to be those guys. So some data still to accumulate. Question?
Yeah, I still have a question about the rhizobium and the plants that we coppice here, such as Gliricidia and pigeon pea and sunhemp and things like that. How does that work?
Those are all legumes.
Yeah, so I may have missed it, but does it drop off in the soil and just get worked back into the nitrogen cycle, or?
Does it drop
I
off?
mean, the rhizobiums on the roots.
The rhizobium form the nodules,
Yeah.
and so they're contributing protein to your plant.
But what about surrounding plants?
Probably not, because where's the connection? What is the connection between one plant and another? What other organism do we have to have?
Fungal.
Mycorrhizal fungi. So if we've got, say, Gliricidia, not a very good representation of it, that's bush, in case you didn't know. So here's our root system. And we've got some great nodules on there and really good nitrogen fixation. If we are already mycorrhizal, or if that plant becomes mycorrhizal, it will form an association with some other plant, perhaps. So maybe your corn, maybe your soybeans, whatever else you're trying to grow. And those mycorrhizal fungi will move some of that nitrogen from this plant into that plant. And maybe other back and forth. It's the mycorrhizal fungus that moves material from one plant to another. Question?
It's common practice to take your nitrogen-fixing branches, all the leaves, and put them around your the plants you want to benefit. Do you like that practice?
As long as you've got the fungi to chew up and break down those woody materials, the bacteria, to chew that up, because then where is that plant material? It's going to be decomposed by that bacteria and fungi. As long as you've got a good food web, great stuff.
And it's that specifically, those legume branches are better than, say, some other one branch?
Well, any legume ought to be just about the same. You ought to see an improvement in the nitrogen content.
And other, say, woody material?
Well, because the woody material in these plants, okay, let's go back. Woody material typically has a carbon and nitrogen ratio of 300 to 1, 500 to 1. That's woody material in a non-rhizobium or a non-nitrogen fixing plant. When we're looking at something that fixes nitrogen, now what's the seed-in ratio of the woody material in a legume plant, a leguminous plant? It's 30 to 1. It's bacterial food. When we're looking at the leaves of a legume, so this is the wood, the leaves of the legume have a seed-in ratio of 10 to 1. They are just like meat. Could we be putting the leaves of those legumes into our compost pile and getting faster decomposition, lots more heat in our compost pile by putting leguminous material into our pile? Absolutely. You don't like manure? Use legumes. So those of us who are vegan, we make our compost piles with legumes.
So in the free living fixation, is that where you get access or brown sugar on your soil? Is that what the results they're seeing to encourage that nodule formation? Is that bacteria is moving that versus sugar?
Only if you've got free livers.
Only if they're there.
Only the free living nitrogen fixers. Yeah, so you have to know that they were there. So could you put molasses or could you put some sugars into your soil to encourage the free living nitrogen fixers to get going? Let's do that try. I don't think anybody ever has. But I haven't kept track of that over like the last 3 or 4 years. I haven't really.
I just read a lot of literature encouraging adding molasses to the soil or spreading your plants or increasing that bacteria or whatever. So I was kind of wondering.
Yep, but I have to have data. Yeah, put that in there, put some sugar in, and that should work better. Not data. I want to know how much nitrogen fixation that we have.
I've been told that like when you coppice the Gliricidia, that supposedly the roots release. Maybe I'm misunderstanding.
Now here we go with if you cut the top part of the plant, it causes the roots to fall off.
No.
When you cut your hair, does it cause your feet to fall off?
No. So that doesn't kind of make sense.
It doesn't make any sense. Okay. Because the Gliricidia needs to now regrow. And it needs all of its root system in order to resupply and get the nutrients back in and start regrowing above ground. So I want some solid data that the plant is healthy and is dropping roots. Never seen it. Now, if your plant is sick and dying and not really well, and you crop is the top part, yeah, the whole plant dies. So now does the root system fall off?
Yeah. They were dropping nitrogen on roots. I'm not sure how it's supposed to.
Yeah.
That was my.
I want to know the mechanism there. Explain to me why your plant that has gone through all of this effort.
I pictured the nodules falling off for sure.
Yeah, so most people are saying that. Ask them the gory details. Okay, how exactly does that work? Because you never know. Maybe they do have the data, and I just haven't read it.
So it's more about the nitrogen in the leaves, you would say, in those systems than the actual sloughing off of, or the root nitrogen, because apparently that's an urban myth or something.
And yeah, we got some silliness out there on that particular one where people observe that when animals graze and you get a big flush of nutrients out of that root system, but we don't get any root death. And then nutrients start to happen, the plant starts to regrow. Well, if you regraze that plant too fast, then this plant is like, oh, yeah, I'm gonna put the flush of nutrients out, but this is kind of painful. I'm using up all my root reserves. Starts to regrow, and you come in and graze too soon. This plant is really getting stressed. It's really getting unhappy, unhealthy. You know, the root system is very susceptible to disease attack. The above ground part of the plant is very susceptible to insect pests and fungal diseases. And so as this plant is just like, I'm trying to regrow, and you come along and graze it again, oh yeah, you'll slough your root system because the whole plant's going to die. So it's not a normal response of a healthy plant. But we have a whole group of pasture people that are trying to make the point that you should not come in and overgraze. You got to let that plant recover. And so they've gone a little bit on the trying to scare people too much. And you know, if you come in and graze your plant, you're going to cause all the roots to slough off and bad. Yeah, it's bad. And it doesn't cause your roots to slough off if the plant's healthy. Have not seen that. So, data. Where's their data? Anything else? I was thinking of something, but we'll probably get to it. So, it's not like we know everything yet. There's way more to be done in looking at some of these interactions. So now I want to, since we've been talking about all these nutrient pools, we've been talking about this system and how it works. When we're looking at soil, what's the right form of nitrogen? Now, standard ecological answer, it depends. What plant are you asking this about? Are we talking about an annual or are we talking about a perennial? Right form of nitrogen for fungi. Right form of nitrogen for bacteria. Protein. What's the right form of? So, in the world of chemistry, what are the nutrient pools that chemists talk about? And I think it's probably really important that you understand this particular problem, because you're going to encounter people who try to get you all confused when they're talking about these different nutrient pools. So total nutrients or total extractable nutrients. Some people use that word, some people don't. This is not normally reported on your soil chemistry test. You are never told about this on a typical soil chemistry test. Not one piece of information about this. Most of the time you can go and find this information. It's in the USDA Natural Resource Conservation Service. It's in their database. So most parts of the United States you can go find what your total nutrient concentration is in your soil because it doesn't change much. As your sandstone clay from your bedrock break down, we are constantly replenishing this total pool, so it's not changing much. Until you run out of sand, silt, or clay in your soil, it's not going to change. It's constantly being replenished. How do we do total nutrients in our soil? It's a pretty nasty process. Typically, when you send your soil sample into your laboratory, or compost or anything else. The first thing that they're going to do is dry that material down. Well, guess what? You just lost some nitrogen. You just lost a little bit of phosphorus. Okay, but not much, right? Sometimes it is a lot. But the next thing that they're going to do is remove all the large chunks of organic matter from your soil. So they run it through a sieve and they're going to remove all the large pieces of organic matter. Wait a minute. Isn't that your organic matter? Doesn't your organic matter? And they just threw it out because it's too big for them to deal with their grinder. So already, aren't we kind of biased on underestimating your organic matter in your soil because they just threw a whole bunch of it out? They're going to take the rest of that soil and they're going to grind it up to a micrometer size. So typically somewhere between 1 and 5 micrometers size. Grind it up with a wily mill. And now we're going to take this material and add it into extremely strong acid. So we're going to go with 24 molar hydrochloric. Really nasty. Everything is supposed to turn into a liquid form. And guess what? Not everything does. So how much is left behind in the little test tube? Oh no, there's nothing much left. Much? Much? How much is much? There's stuff left. So how important is that stuff? Well, okay, but it's ground up, solubilized as best we can. And now we're going to take that liquid, because it's now all been liquefied as much as we can. And we run this through a gas chromatograph or atomic absorption. Different kinds of machines to look at total concentrations of your nutrients. Or best, mass spec. Mass spectrophotometer. We're getting more and more finely resolved information with each of these different systems. So you can be sending your sample into labs that have mass spectrometers. You're going to get the most precise data. Well, but then it's expensive. It's probably going to cost more than other labs that do not as precise information. Most soils don't change much with time, so really do you need to be doing this very often? Maybe once every 100 years. If you do something really weird to your soil, maybe a big disturbance happened. But as long as you're still dealing with the same sand, silt, clay, probably not going to see much change there. What if you add a bunch of organic matter? Okay, then, well, as long as it's organic matter that they're not going to sieve out when you send it to your soil chemistry lab. Total nutrients tells you what's actually there. Yeah, but you're getting nitrogen, total phosphorus, total calcium. Is that really what we need to know? The next thing that they're typically only going to tell you a little bit about on a soil chemistry test are exchangeable nutrients. And so when we're looking at exchangeable nutrients, the name put on this is typically base saturation. When you run a strong base through your soil, how much of the nutrients that are held on the surfaces are going to be knocked off? And so we're going to measure that increase in the concentration of those nutrients as we knock those nutrients off surfaces. So this is all surface bound. This does not tell you about what's in the bacteria or fungi, protozoa, nematodes, microhypopods, mycorrhizal fungi, roots. Doesn't tell you anything about that. Doesn't tell you anything about what's in the organic matter. It doesn't tell you anything about what's in your sand, silt, and clay. Only what's surface bound. And typically the only nutrients they're going to talk about are calcium, magnesium, potassium, and sodium. So we're talking base saturation. These are the nutrients that Albrecht dealt with. So Albrecht was the head of the Department of Soil Science at the University of Missouri for many, many years. He started doing studies where he was looking at these exchangeable nutrients and showed that if you're lacking the proper balances of these nutrients, animals eating the plants grown in those soils, we're not going to be healthy. And the work that he was doing annoyed all of the chemical companies. And so he was quite unfriendly, invited to leave his job as head of the Department of Soil Science at the University of Missouri. He was told to retire or else. He retired. So, what he showed was that in most of our soils in the United States, in the mainland, anywhere we're dealing with montmorillonite, a certain kind of clay, so it's montmorillonite clay, the ratio of calcium to magnesium needs to be the percent calcium compared to the percent of magnesium needs to be 7 times more calcium than magnesium. That's a montmorillonite. What's your soil clays here? Well, it's not montmorillonite. So when we're dealing with most soils here in Hawaii, we're dealing with volcanic clays. And typically when we're looking at the andesites that most of the Hawaiian Islands have, so what came out of the lava, and as the lava breaks down into sand, silt, and clay, the clay that you've got is andesite, extremely high in calcium. There should be no reason at all, ever, to add calcium to any of your soils here in Hawaii because you have excess, you got too much. Notice how when your soils get real dry and you're walking across them and you put your foot down and it kind of does the moon dust thing? That's telling you got too much calcium. You don't have enough magnesium. How are you going to put those 2 nutrients back into your soil? Compost.
So when I've done really testing around here, most of the time I come up with too much magnesium. The soils are really tight and chewy and sticky, and then if you put calcium in it, it really opens it up, and it's not so awesome like that.
And how long does that stay nice and open and not tight and looking good?
Well, when I retested, the numbers certainly come back a lot better.
It doesn't usually last too long though. So in my experience,
Oh, no,
I do.
I usually don't retest for at least 6 months. And so they get better over time. That's what I'm saying.
And so you are probably on something other than andesite. So what is your clay type then?
It's a silty clay, I mean, that's all I've ever really heard it's called.
Yeah, so it'll be fun to investigate that and figure out what's going on. But you need to really be looking at the kind of clay that you're dealing with. If you're on a montmorillonite clay and you don't have enough calcium, okay, you're going to have soils that tend to glom together. Your clays stick really badly. So are you actually on a volcanic clay or are you on something else? Get your soil up to 50% moisture. And what you want to do is take that, some of your soil and kind of mix it together with your hands. And if it doesn't ball up, if it doesn't stick together, your calcium-magnesium ratio is probably not too far off. But if it balls up just like some dough, you want to roll that between your hands. And the further you can roll that soil out, the more you have a calcium problem in your soil, and you would want to probably get calcium back in there. I would like to see you add in as compost and not as calcium carbonate. So, because we really don't need to be leaching that calcium out into your rivers and lakes and streams. This is another case where work with soil, with people. They've been sending their soils into University of California, Davis, for example. And for 32 years, they've been sending their soils into UC Davis. For 32 years, every year, they were told 3 tons of lime per acre and a half a ton of gypsum. They go and retest this the next year, and it's exactly the same thing. Their calcium to magnesium ratio is just as bad. So more lime, more gypsum for 32 years. And so he comes to me and he says, you know, I really don't think this is working. You're absolutely right. I don't think you're getting much benefit from this. So what do you do instead? A real good example of a time where you need to put organic matter and biology back into the soil. And when we did that with this person, we got our calcium magnesium ratios perfectly balanced. They send their samples into the soil chemistry lab, and now it says you don't have to add anything. One application of really good compost and or compost tea is what we're seeing. So is every place on the planet the same? Absolutely not.
So what I've heard about those extension labs, a lot of times they'll just look at your soil type and they never even actually do that test, and that's why they keep sending you the same thing every year, because they never even really look at your stuff.
Go talk to the guys at UC Davis, and they absolutely swear they actually do the analyses.
I mean, I've seen some of those stuff come out of those labs, and they may even give you a number, but they don't even, like on the trace elements here, you send your stuff in to UH, you can get some numbers, but they don't even say what they are, high, low, nothing. They don't ever address it at all. It's ridiculous.
So
Why pay for a test and there's nothing that comes from it?
don't use that lab, would be my suggestion to you. Go to someplace else where they can't explain it to you. And where they will give you an idea of what the normal expected values are for these kinds of materials in your kind of soil. So let's keep going on this. Total exchangeable nutrients, that's what we're looking at, is calcium, magnesium, sodium, potassium. Now, please recognize that different labs use very different extracting agents for these exchangeable nutrients. We can use a really strong extracting agent. So if your lab uses a Melic 3, you're going to be given a much higher number for calcium, magnesium, potassium, or sodium as compared to a lab that does a Bray 2, or a lab that does an Albrecht extract. You have to have some understanding of what your laboratory is actually doing, and I do strongly object to laboratories that don't give you any information about what extracting agent that they're using. How many different kinds of extracting agents are on the soil chemistry books for extracting exchangeable nutrients? 650. My buddies in Australia, the head of the environmental analysis lab, is the one who looked that one up for me. 650. Which one is your lab using? And if you change labs, how do you now compare? It can get to be pretty difficult. And now what mostly is on your soil chemistry lab report are the soluble nutrients. So what extract are they using to extract the soluble nutrients? Why don't they just use water? Well, because that would be too easy, wouldn't it? So which extraction agent are they using? So I talked to Graham Lancaster's Southern Cross Environmental Analysis Lab, and I asked him, so how many different kinds of extracting agents do we have for soluble nutrients? And he said, probably about 1,000. So which one's your lab using? And are they doing things exactly the same? It makes a difference whether you take your soil that's already been ground, and whether you add that to water and then extract, or you extract that and then add the water. You're going to get different numbers. What's reality? How do you really know what your plant needs? Well, let's do a tissue test. Tissue test is exactly like the total nutrients. We're going to get your plant material. We're going to grind it up to a real fine level. We're going to use a really strong acid, try to solubilize everything, and run it through mass spec, or GC, or atomic absorption. And now you know what's actually in your plant. We can do a really good job with plant tissue, unlike soil, where we got all kinds of yuck. So that tells you what your plant actually has. In all of these soil nutrient pools, which one of these pieces of information tell you what your plant is actually going to take up? None of them. There is no correlation at all whatsoever between soluble concentrations. I don't care what extracting agent you're using, there's no correlation with this and what your plant actually is going to take up. There is no correlation between exchangeable and what your plant actually takes up. So pray tell, why are we doing these? In what way are we getting information that's useful to us? Or are we getting information so some soil fertilizer guy can scare you? Oh my God, you've only got 50 parts per million nitrate and your plant is 800. Oh, you're going to have to put in 700 and blah, blah, blah. Tons of, or pounds of nitrate into your soil in order to be able to make a normal crop yield. I can show you soils where our phosphate level is down around 0.1 part per million. Oh my God, your plant, it better have at least 1 part per million phosphate or your plant's just not going to grow. Go look at the plant tissue. We have excess phosphate inside the plant. This means nothing when it comes to what your plant's going to take up. Go to any old growth forest and look at the nitrogen, phosphorus, sulfur, magnesium, calcium, sodium, potassium concentrations in that old growth forest. Have fun with your soil chemist guy. Send them into one of the old growth forest samples and watch them go ballistic. You don't have any nutrients, this is just terrible, your plants must be dead. That explains why it's an old growth forest. Where we are sequestering every year more nutrients in those trees than we are sequestering in any crop that you want to grow. So why are we doing these nutrients? Why are we looking at this? Probably the only thing I really want to look at is that exchangeable nutrient, because I kind of like to have an idea of what that ratio of calcium magnesium is. So I think that may be pretty useful when we're starting in a new soil. I don't know if we've got problems there. Do we have our clays in proper order, or are they probably just totally out of whack? Because when we look at these calcium-magnesium ratios, what we're really trying to figure out, and really what Albrecht was trying to understand, was whether our clays are collapsed. Do they have good structure? Or are they flocculated? Do they have good structure? If our clays are collapsed, we lack calcium. Our clays are going to be stacked up. Nice columnar clays. And here's our next stack of our clays, and our next stack of our clays, and more clay stack, and another one, and have some fun drawing this because it's like plates sitting, waiting for you to wash them on the edge of your sink. They're stacked up. You can't get oxygen, you can't get water, you can't get organisms growing in this stuff at all. There's no space. It's collapsed. How easy would this be to compact? Very. So we may have to get that calcium back in there. So add calcium. What's the best way to add calcium? So I do have an experiment that I'm going to show you. But if we add that calcium, it causes each one of those clay platelets to repel each other. So the next clay platelet, the next clay, the next clay, and you can see how we're building structure. So now there's plenty of space for oxygen, water to move through our soil. There's condominium housing for our bacteria. Clays and bacteria, clay colloids and bacteria are about the same size. And so now we have places for our bacteria, we have a little bit of water being held in here. Things are better for everybody concerned in our soil. We have too much calcium, we gotta get magnesium back in there. So let's understand what that ratio is. But I really don't see a whole lot of reason to be looking at the other soluble nutrients. Or maybe when I buy a property and I know that it has been hammered with pesticides and inorganic fertilizers and I don't have any decent biology in there, then is a good time to be looking at your solubles and your exchangeables and get an idea if something's just so out of whack that we ought to put more compost in than we might have planned that we need to be putting in. Or maybe it's a good time to put in some rock phosphate or what's the form of calcium you ought to be putting in.
Do you have, you spoke of calcium magnesium ratios in play and the proper ratios. Do you have an idea of the andesite, proper calcium magnesium?
Yeah, I know I've talked to Bob Schaefer about this, and he's one of those people who's worked a lot on these andesite clays. And he grows coffee over in Captain Cook. And I would have to go and remind myself what that proper ratio is. I think it's like 5-to-1. You can't put as much calcium into an andesite as you do with a montmorillonite clay. But that might be a good question to ask your soils person. What is the proper calcium magnesium ratio for an andesite? Because it's quite different from montmorillonite. Albrecht worked on montmorillonite soils. The only thing he ever worked with were those soils in the middle part of the United States. Everything is montmorillonite-based clay in that part of the world. Everything in the western half of the United States is on basalt. And the clay is montmorillonite. So it works for most of the U.S. What if we get up into the Hudson River Valley in New York? And it's not montmorillonite, it's kaolinite. So you can't get that much calcium into a kaolinite, but it does have an infinity capacity to grab phosphate, potassium. So that's always fun to point out to people. Illite, Illinois, illite, different clay type. Again, you can't get 7-to-1 in an illite. So as you might be working in different parts of the island or in different parts of the United States, you might want to go to the soil survey and get some of that information, the major clay type on your soils in your part of the world. So really, what do we have to have in our soil to move nutrients out of the total pool into the exchangeable, into the soluble, and then as our plants, our microorganisms take that back up, we'll be moving them back into these different pools. It's biology. Everything's done by biology. If you want to increase these pools, you've got to get the microorganisms doing their jobs. Another way of looking at this, the total amount of nutrient in your soil. Well, we grind concentrated nitric acid. We're going to get this much of the total. But we only use 10% hydrochloric or nitrate, we only get this much of the total. So you can start comparing these different extraction methods. What's your lab actually using? If we use an LI3, we're doing an exchangeable. So you're going to get this number. What if you use a grade 2? What if you use an Albrecht? Okay, now let's go into the soluble pool. If you use a Colwell, a grade 1, or an also, this might be useful for you if you've had soil samples done in different laboratories. If you really want to understand where, a universal buffer, or the Morgan, the Reams lab, what are they doing relative to other things? But soil chemistry can drive you crazy pretty fast. Don't you agree? Here's a little experiment, which is because I was kind of concerned with the answers I was getting from different soil chemists. It just didn't make any sense to me. What really holds calcium in your soil? When you add calcium, what is it that holds that nutrient in your soil? So we had a sandy loam soil, mostly silt, sand, not a lot of clay, but it is a montmorillonite clay. So this is going to work for Oregon, but we might have to repeat this for Hawaii. We went to the riverside and we got this sandy soil and we removed all the organic matter by flotation. So we mixed it with water, all the organic matter flows to the top and we take that off. And now we're left with just the sandsill clay. We sterilized that sandsill clay because we didn't want any biology. But what really holds calcium when you add it into a soil? Is it the organic matter? Is it the sandsill clay? Or is it the biology? And we wanted to ask that question. So in the first treatment, this was sterile pyramid sandsill clay, repacked in the same bulk density as the original soil. Put a little bag of calcium carbonate, oyster shell, ran a liter of water through here, and we could then collect the water that came out the bottom and determine how much of the calcium that leached from that oyster shell. And so we could measure that, 300 micrograms of calcium on average in general, a little bit of variability. But 300 is what leached into our soil column. How much of that calcium could we measure coming out the bottom? And I just loved it that we could measure 315 micrograms coming out the bottom. So tell me, does your sand, silt, or clay hold on to the calcium you add when you add it into your soil? No. The exchange capacity is already occupied. It's been occupied for like, the last million years. So just simply adding calcium into that soil, calcium carbonate, is probably not going to make any difference at all. And we just end up with most of it heading towards your drinking water, your groundwater, your soil water. Not really impressive there. So then we had that same sterile parent material, to which we added 5% organic matter. The organic matter was a compost that we autoclaved, sterilized, no life. But we're adding the organic matter back. So same little bag on the surface of the soil, ran a liter of water, so 300 micrograms go in. We collect all the water coming out the bottom. Yeah, a little bit of water was held in here because it was about 5% organic matter. But of the water that came out the bottom, how much calcium came through? We could detect all except for 2%. 2% of the added calcium actually stayed in there, so whoop-de-doo. Organic matter, sterile, does not really do the job either. How about bacteria? Bacteria, we added bacteria inoculum back to the sterile compost material, that organic matter, 5%, water, ran it through. How much of the calcium, 300 micrograms that we put in, how much of that could we detect coming out? 95%. Soil, let's see, the organic matter, sterile organic matter holds on to 2%. Your bacteria hold on to 3%. Whoopee. Not going to be doing much to fix your soil and getting your clays to flocculate. So now we put a fungal inoculum into that same sterile compost. 5% of that mixed in here. Ran 1 liter of water through that little bag of calcium carbonate oyster shell. And how much of that calcium, 300 micrograms that went in there, how much could we detect coming out the bottom? Nothing. No detectable calcium. So tell me, what holds on to calcium in your soil? What's the mechanism?
It sticks on there, calcium oxalate.
The oxalate, yep. So it's biology, it's not what's going on with your chemistry. So we've talked about this, how much calcium is added into your soil when you put it in compost? So this is kind of a fun one. Here's some typical soils. And we're looking at total calcium. That's not really impressive in the soil, is it? These are agricultural soils, and they've been pretty damaged over the years, because these are not good levels of calcium in the soil. What's in the exchangeable pool? So most of the calcium in that sample is sitting on the exchangeable surfaces. Run enough water through here and you're losing all your calcium. Here's the soluble, available calcium. It is going to be leached out if you run water through this sample. Now this is grapevines. This is kiwi vines. Here's our compost. So we put a ton of compost back into our soil. We are going to be giving ourselves a good many years of calcium back into that soil. If we've got the biology, it's going to be made available and start flocculating on clays nice and quickly.
Just wondering about some of these composts that are made from plant material that's really low in calcium. And, you know, it's sort of like you put it in there, you compost it, you're hoping that you're going to get more out of it, but I mean, garbage in, garbage out?
Yeah, probably. If you really have plant material that has no calcium, but be aware what pool are you looking at when you say the food material has no calcium. Total or soluble? And so you gotta go back and look at that. Because I think most vegetable material has a pretty good amount of calcium in it. Because this is compost made from plant material. And look at the calcium in there.
It's pretty variable. I mean, those tissue tests that I've done can be 10 times different. You know, same cultivar, really.
Yeah.
And it's just, you know, how's it growing?
How's it grown? So lots of work yet to do. Now that was the calcium in the top 10 centimeters. And I love it when you go to a soils person and they're going to base what you have to add back in only on the top 4 to 6 inches depth of your soil. They calculate what's available to your plant only on this depth of the soil. Do you have calcium deeper down in your soil? Oh, calcium falls off really fast. You got lots here and you don't have too much there, and that's almost nothing. And this is data. Could I have data, please? Let me show you the data. This is the top foot of your soil. There's the second foot, there's the third. How much difference? Is there like no calcium down there in 3 feet? There's plenty. Look at that one. Look at that. 12 different soils. How many this is your soil? A little bit of your calcium is leaching down into the deeper depths. What if you put on the amount of lime that they would calculate for you, has to be added back into the soil? And you put your plant into that soil and look what it hits at the third foot. Whoa! You're going to cause some damage. We don't have a lack of nutrients at depth. As long as we don't have compaction layer, as long as your roots can grow down through there, we don't have to be adding calcium back into this soil. Yeah, we gotta have biology to make that calcium available to you, but this is soluble calcium in 12 different soils, or 11 different soils. So where does this come? That we don't have nutrients deeper down in the profile. Absolutely bogus. Do not do all your calculations on the nutrients that you have in the soil just on the top 4 to 6 inches. You're leading yourself astray.
So would that plant then have a problem with taking up too much calcium once it hits the 3 foot level?
I think this is probably going to be a salt problem. Because especially if you added calcium here and then it hit this as well, a little bit scary. So, how many of you seen this one? The only thing that determines the availability of nutrients to your plant is the pH of your soil, right? If you have no biology, this is the only thing you got. So I love the fact that soils people, or the world of fertilizers, I pick on the right person here, say that your soil should be at a pH about 6.5 to 6.8 right here. Well, molybdenum, is molybdenum most available at a pH of 6.5, 6.8? No. If you had a molybdenum problem, would you have to up your pH to 8.5? How about copper and zinc? Are they most available at 6.5? No. They're way more available if you get a little more acidic. So now if you've got copper or zinc problems, when you have to drop it down to a pH of 5.5.
It's about as much there.
Boron. Is it most available at that level? It's well, you know, you could have your pH at 7 or you could have your pH at 5.5. Why do we pick on 6.5? So manganese, is it most available at 6.5? No. Shouldn't you have acidify your soil? How about iron? Whoops. You know, how about magnesium? How about calcium? It's most available up here. How about sulfur right there? Potassium. Well, what's the only nutrient in here that is actually most available at a pH of 6.5, 6.8? Phosphorus. Look at that cute little bird right here at a pH of 6.5, 6.8. Nitrogen, pretty well even across the board here. So why is it that phosphorus wins? In the fight between your dollars, why do the phosphorus guys win?
Most expensive.
Whereas if we put biology back into the soil, throw this all out the window. Never waste your time with this one again. Because it is the biology that makes the nutrients available, not the pH of your soil. Because if we really followed this, we'd be bouncing the pH in your soil up and down and back and forth all the time, trying to figure out what your plant requires most at this instant in time. So, lab reports, can we deal with this? How tired are you all getting? Well, let's dive into it, because it's really crazy. I give you a soil report, a chemistry report, from a lab in Australia that we work with. Because I love the fact, and now many soil chemistry labs are starting to follow this as well, where they actually tell you the extracting agent that they use. So now you can really understand the difference in these different extracting methods. Soluble tests using a morgan 1 extract. So it's a little bit more capable of extracting soluble nutrients from your soil than water. So we're going to get a higher number. So we're looking at calcium, we're looking at magnesium, potassium, phosphorus using a Morgan extract, phosphorus using Bray 1, using a cola, using a Bray 2. Okay, which ones tell you truth? Of all these ways of measuring phosphate, which is the true concentration of nutrient that's available to your plant?
What's the closest one?
The highest?
Well, why is iron the best? And in what is the relationship between any one of these things and what your plant actually takes up?
Like,
So I almost giggle at these. Look at all these different extracting agents. They're all extracting soluble tests. They're all telling you about soluble phosphorus. Which one's true?
They all are.
yeah.
Or none of them? How much is your plant going to actually take up? This particular plant, absolutely no limitation with phosphorus. And yet when you look at what's typical, well, are we dealing with a heavy clay soil? Or are we dealing with a medium, a sandy soil, or excuse me, loamy soil? If I had enough space, I would put on the sand, and this would drop by another, just about the same, so from 15 to 12, from 12 to about 9. What kind of soil do you have? So which is the right comparison? Does your soil chemistry lab tell you what's normal for heavy clay soil? For a loam soil? Or for a sand? Very different values. When you start looking at whether you're in a heavy clay or a loamy soil, sandstone clay. So which one is true? I think none of them are. Our plant had no problems when you look at the plant tissue test for phosphorus. And yet if you followed these tests, wouldn't you have said that, oh my God, I better put phosphate fertilizer in here? Because the biology in the soil did the job. You don't have to worry about was actually chemically available or not and which of those tests that you did. So here's nitrate. So So look at the nitrate level as being kind of high. Now that is an interesting piece of information because really what's this telling you? What kind of weeds are you going to grow? And you're going to be growing a boatload because this is way too high a level of nitrate. We're looking at ammonia. That would tell you that you're going to be hurting. So let's not pretend that we're going to, or this would say we're not going to be able to grow any of our perennial plants. And yet our perennial plants were doing just fine. Looking at our sulfur. Oh my gosh, we better get some more sulfate out there. No, there was no problem with plant tissue test, the sulfate level. pH. Now that might be interesting because if your pH, the general pH of your soil is 5.29, what might you suspect right off the bat? We're a little bit on the anaerobic side. Could be a concern here. Conductivity, so we're looking at that microsiemen thing. Look at the desired levels. If what your soil should have is 200 microsiemens, or if you're in a slightly sandier soil, or very sandy soil, it does vary. You better not be putting an engineered soil out there that has 10 decisiemens per centimeter square. Organic matter, looks pretty good. Now we're moving into the Albrecht, exchangeable. So our calcium, I like it, that it gives you whatever way you want to calculate. Which way do you understand? I like first per million. So we should have 3,000. We've only got, almost 2,000. Yeah, we're working a little over calcium. Magnesium, a little bit high, isn't it? So you can be looking at, this data would suggest to me that my soil needs a little bit more calcium in it to flocculated clays. So I'm going to get some compost in there to give things a little bit more ability to fluff out. And here's something that's new in the world of soil science reports, or soil chemistry reports. They're starting to add other things. They're putting aluminum, they're putting hydrogen into the air. Did Albrecht, in any of the studies that Albrecht did he put aluminum into these calculations? Did he put hydrogen into these calculations? No. So now the calculation of your percent calcium, your percent magnesium, percent base saturation is messed up by adding in aluminum and hydrogen. How do you account for that? What if you had really high levels of aluminum and hydrogen, then these ratios are going to be, you can't use them to do your calcium-magnesium ratio. Why are they doing this? Put other stuff in there. Because Albrecht did just these 4 elements, nothing else. I want them out of here. Calcium-magnesium ratio. Is this a little painful? We need 7. We've got 0.8. It's a little scary. Buffer pH. This is just a measure to try to convince you that you need to buy lime. That's all that's for. So the lime industry has done a really good job trying to convince people to buy lime. They're quite successful. Look at that iron. Should you be concerned about this level of iron? Doesn't that look like you've got a toxic level of iron? Yeah, don't worry about it. It just means your soil was anaerobic in the past. And it's now become oxidized, you've got high amounts of not plant available iron in your system. Better get the biology back in there. Do you really need to worry about anything else? Zinc looks a little painful. But if we put compost in there, we're just fine. So, if you want to have fun with your soil chemistry tests, bring them on in and we can spend a little bit of time going through them if you'd like. Pretty straightforward. Okay, soil biology. Going on. This is a soil food web report where we're looking at active bacterial biomass, total bacterial biomass. What's the difference between active and total? Total is everybody in the room. Active are the ones of you who are still paying attention to me right now. The ones of you who are sound asleep, you are part of the total population, but you are not part of the active population. So when I used to give introductory biology lectures, beginning, I'd come in and I'd be setting up, and all 600 students would come into the room, and they'd be fine in their place. They're all performing their function of coming to class. 100% of the students in the class were active. So they all settle down. I start my biology lecture about 15 minutes into the talk. How many of them are still active, performing their function? How much of them back there writing notes and spit wads going and airplanes and, reading the newspaper, not performing their function. It's a whole large percentage no longer performing their function. They're part of the total, they're not part of the active. 45 minutes into the lecture, just before we're going to finish up, I always figured it was the front 20 that were still with me. So what's the percent activity when 20 out of 600 are still actively listening to the lecture? It's pretty depressing, isn't it? Okay, are those organisms out there capable of coming back to life? How do you know? The bell rings! And they all out the door. They were all capable of coming back to life fairly rapidly. So there's your active, and there's your total. So how much of this population is actually functioning? So that's what we're looking at here. We're looking at active fungal biomass, total fungal biomass. So in this biomass, is my fungal biomass more active than my bacterial? So I'm going to be growing more fungi. Over time, I would expect this number to go up faster, and this number would go up. Now, looking at my total bacteria, do I have a good number of total bacteria? It's looking pretty nice, isn't it? So it's, way above minimum level. But that's what this value is, minimum. I have at least. My total fungal biomass. Wow, that's really good. It's way above minimum. And now let's look at the balance of fungi and bacteria. This is a fungal dominated soil. Is that what I need to grow the plant I'm trying to grow? Wait a minute, what plant am I trying to grow? Oh, banana. Banana needs to be fungal dominated. So, woohoo, this is good. If this is a compost I'm putting in, this should really help move things towards the direction that my banana might need. Do I have good guy fungi or bad guy fungi? These are good. Wow, really nice. So protozoa, do I have enough protozoa to do nutrient cycling? Oh yeah. 28,000 I only need 10. 15,000 I only need 10. So we got some good nutrient cycling going. I don't have very many ciliates, so life is wonderful. There is no anaerobic signal here. What do my ciliate numbers have 2 more zeros after there? Scary, huh? But no, we're in good shape. Now look at my number of nematodes. I got 65 of them. Do I have good nematodes or bad nematodes? And that's where the next page comes in. Oops, going the wrong direction. So we list for you, this is bacterial feeders. We list for you the different genera of bacterial feeding nematodes. We got good diversity. We got lots of bacteria feeding nematodes. So this is looking really good. We want 6 species. Fungal feeding nematodes. Yeah, we're a little lacking here, aren't we? We've only got 3. We want 6. Because of this compost, it's like one of the only things we can really improve on. We don't have any root feeders in there at all. And we don't have any predatory nematodes in here either. So minor improvements would be possible. So we usually calculate for you how much plant available nitrogen supply is going to be produced by your predators eating your bacteria and fungi. And so the supply to our bananas, if we put this compost in and we maintain these numbers of organisms in our soil, we're going to be releasing between 150 and 200 pounds per acre of plant available nitrogen. More than enough to grow our bananas. So the question really becomes is, once you add this compost into your soil, what happens to all this biology? Because the compost sure looks nice, doesn't it? This is good compost. So let's go on to another sample. And now it's up to you to tell me, is this good compost or not? Why not?
Really high sewage.
Yeah, look at those ciliates. Isn't that frightening?
High bacteria, low fungi.
Look at the total and look at almost none of this is actually active. When we're looking at activity here, it is only aerobic activity. And so most of those bacteria are probably anaerobic. Anaerobic. No aerobic flagellates and uni. We don't have any fungi in here to speak of. So all of our filamentous fungi have been wiped out, anaerobic. So that easily, that simply, you can tell whether this is good compost or not.
Where it says equal fungi to bacteria in the top corner, is that what's desired?
That's what's desired. So for asparagus, well, that's not right, is it? Asparagus, we would actually want it on the bacterial side. But not asparagus, we do not want anaerobic conditions analysis either. Really like this is an actinobacteria for asparagus. So looking at ours, and look at this, we got rotating nematodes in here. Now, we have another sample, and this is a tea. So I want to go ahead and pass that one around and split it into groups because David wanted us to look at this really quick. So this is the current report form from Soil Food Web. And you'll see that it's a little bit different from the one I had up there. So pass it on around. Everybody got theirs? So is this a good tea or not? It just says T1. We don't know what plant. So we're going to have a hard time interpreting this if we don't know what plant you want to put this on. So what do we need to be approving in this soil? So let's just look at it generally as is this a good tea or not on average? And so I'm going to generally be thinking about this in terms of a equal biomass of fungi or bacteria. So this could maybe be going on almost any plant. So does this have an equal biomass of fungi and bacteria? Doesn't appear to. So active bacteria, active fungi. Is this tea becoming more fungal or more bacterial? Yeah, so it is moving more fungal. But look at total bacteria and total fungi. Is it balanced? So if we're going on a plant that needs equal biomass of fungi and bacteria, we're kind of hurting. It is becoming more fungal, but very well isn't what our plant needs. But that's why we need to know your plant. Do we have good fungi in here? Yeah, look at those diameters. So, going down to our protozoa.
Numbers are good.
Looks really good. We've got good protozoa in this tea. We're not outrageous with the ciliates, so this is looking really good. Nematodes, a little low. It is really hard to get good nematode numbers in tea. It's possible, but you have to have really good nematode numbers in your compost. So I would suspect they didn't have really good nematode numbers in their compost. Now look over at the right-hand column where it says nematodes per mil of tea. We have some bacterial feeders. That's good, but only 2 species. Maybe a little lack in there. Fungal feeders, we got some of those. Pretty good. Predatory nematodes, that's excellent. To have predatory nematodes in your sample, that's really good. So what few nematodes they've got looks pretty good. So total fungal to bacterial biomass ratio on that bottom row, 0.4. You know, if we put this on the soil, what kind of plant would we be selecting for? Annuals, we're going to be down there in the brassicas, you know, maybe some early vegetable species. Is that what we want? You know, that's what we got to know the plant that you're trying to put this on. At least when we're looking at active to total fungi, active to total bacteria, we don't have an outrageously high set of activity, so we're probably not going to drive our compost tea anaerobic very rapidly. We are going to be growing more fungi and bacteria, and so when you convert this into plant available nitrogen supply, good predators, so we're going to have lots of nutrient cycling. This could be really good if our plants needed to be zapped a little bit in terms of getting nutrient cycling up and going really rapidly. We did have Actinobacterias in the sample, about 9 of them, 9 micrograms. That's a little high for most of our perennials, a little high for any vegetables or row crops. It would be great if we're putting this tea onto brassicas or mustards, kale, crops like that. So this looks like it would be pretty good for those kinds of crops, for fairly early successional, you know, mustards, things like that. The kinds of nematodes we have is good. The quantity's low. But at least we get the inoculum in there, which would be way better than nothing. Okay, so see how I did that? It's really kind of straightforward. No special magic involved. This costs about $120 to do all of these assays like this. Notice that they didn't do the mycorrhizal fungi. So if you do the mycorrhizal fungi, it'd be another $40. And so if you just look at Oregon Food Web, there we go. Yeah, it is the Oregon Lab that did this. So there's their email address down at the bottom. So go to their website, and they've got the prices on this on their website. Okay? So, coming on. Now we got through the soil report. Do you want to know soil chemistry or soil biology? Now, what time of year are you going to be sampling? When we want to go out there into the soil and take a look at what our biology is doing. When we're in temperate systems, this is a typical pattern of activity. Activity, not totals, activity. So in temperate climates, typically in the winter, soil's frozen, not really high activity. The only activity is occurring in that top few centimeters of your soil. If you've got a layer of snow, you're going to have a couple more centimeters of active organisms. But if you compare that over soil profile, not real high. But real good activity is right there. Under the snow. Bacteria, fungi are growing happily, they're doing their thing. And now it starts to warm up, your snow melts, your plant stops pumping out exudates. Bacteria, fungi start growing, pumping out the exudates. These bacteria and fungi are starting to grow really fast, protect your root system. Protozoa nematodes are being attracted into the root system, and nutrient availability, massive, really good nutrient cycling. Your plant's going, yes, yes, go boys, go! And then the plant says, oh, I got all the nutrients I needed to go into reproductive mode. So thanks, guys. It's been nice. See you next year. And that exudates into the root system, turn off. And so activity in that root system then plummets. And in the middle of that hot, dry summer period, we may have no detectable activity in that soil, or again, it may be very, very low. In the fall, when you harvest, and you put the residues onto the soil, and we're getting fall rains, we may have a second burst of activity, decompose all of those plant residues. We want to see the residues sitting on the surface of your soil disappear. The within a month, turned into soil. Well, if they don't get turned into soil right here, hopefully, we're going to have a snow layer. We're going to have some of that residue decompose and turn into soil through that winter period. So when the snow melts and it all takes off again, we don't have any of those residue materials sitting on your soil surface. Typically, those residues that are left over tend to be the places where our insect pests are hanging out, where our disease-causing fungi are hanging out. Don't want any residue come the next growing season. Well, how about here in Hawaii? A little different. Dry season, activity's a little bit lower, but notice quite a bit higher through your dormant season. And as rains happen, so we're coming out of that drier part of the year, your plant is putting out exudates and saying, go boys go, good nutrient cycling. Your plant says, thank you very much, it's been a great growing season. I'm now putting all my energy into finishing up the fruit. Getting that reproductive structure dealt with, and bang, down it goes through that low level of activity through the middle of that dry season. So, differences in different kinds of climates.
How does this relate to when you would test?
So when would you want to test? So I would suggest you probably want to, well, first of all, when you probably want to test is right on in here. Are you getting decomposition of all the residues that are coming down onto the soil in the fall? Are things set up so you've got the right balances for whatever plant it is you want to grow next growing season? If it's the same plant you got in there now, great. If you're doing a perennial, great. You know what that is, but maybe you're going to grow broccoli instead of peas. Or maybe you're going to grow sunflowers instead of tomatoes. So you might want to adjust this, and so you might be putting on this kind of compost, or putting out tea, or putting out fungal foods, or putting out bacterial foods, depending how you want to adjust that biology. And then again, testing in here to make sure that whatever adjustments you did there have actually fixed the problem, have actually made proper adjustments, so everything's set up and ready to go, so you're going to get the most productivity that you want. Be putting a compost tea, typically compost extract on your seeds. So when you're doing annual plants, make sure the proper biology is on your seeds, so when you're planting those seeds, it's going out with the right biology. Annual plants, again, you might be putting a tea or a compost right down that planting row. So you're putting those seeds into something that's going to give them a good, strong, protective layer around those root systems. Okay? Pretty straightforward? Makes sense?
So you need to soak the seeds in the tea?
Yeah, you would soak either in an extract or tea. Sometimes I go for extract, it's a little bit easier to make. And you would typically soak your seeds for about 15 minutes. If your planter can't deal with wet seeds, then dry them down. That biology has gotten in the cracks and crevices in the seed. They're under the seed coat. So drying that seed down is not going to be a problem. So get it dry and on into your plant box they go. So you might have to dry it down. Bare root trees, bare root shrubs, strawberries. Just dip them into your compost tea with the right mycorrhizal fungi, if you don't have mycorrhizal fungi in your soil, and let them soak in that compost tea with the mycorrhizal spores for about 15 minutes. Pull them out, they'll drip dry, go plant them. So the food web. You can take me through here, right, and lead me through and explain everything to me now. Practice. You might want to practice tonight. The basic premise is that all the biology has to be present. We want to know what's the balance for the plant you want to grow. So you might go back to that successional picture. We want the full food web, we want the full diversity. So if you go back to that successional picture, and I ask, what is the proper food web for strawberries? Where do strawberries fall on that successional picture?
early succession.
Where do you find strawberries in the natural world? Is it up here with weeds? Is it with brassica? Is it with your veggies? Is it with a good meadow or row crop system? Is it with shrubs or trees? Shrubs, bushes, shrubs. It's getting late, isn't it? Bushes, deciduous trees, or is it with conifers? Where do strawberries occur in the real world? Understory plant species in forests. So what is the proper ratio of fungi to bacteria if you want to grow strawberries without pesticides or organic fertilizers? Very fungal. We want these things around 10 to 1, 100 to 1, somewhere in there. So why is it in our agricultural systems we keep trying to plant strawberries in soil that we have just methyl-bromided? What's the biology left in a methyl-bromided field? So why do those strawberries not taste very good? Why are they chock full of diseases? Why can you only pick one picking off those strawberry plants before they're dead? Because we keep trying to grow them there. Maybe not too bright. We need to get that fungal biomass so your strawberries are going to be gorgeous. How many years should strawberries be in your field and you can continue to pick them through that whole period of time? How long can you keep a strawberry plant alive?
Perpetually.
Yeah, perpetually. We've got strawberries that are 5, 6 years old. Now, a lot of people, the strawberries kind of get a little bit root bound, or they're putting out so many runners that they let the runners take over and they pull the mama plant. Okay, maybe it's not quite as productive. If we really got the food web right, maybe you wouldn't have those plants. So we got a little more work to do on some of that productivity issue. So you're trying to grow bananas. Where do bananas grow? Yeah, so they're a big grass, so they're on this, they're towards that. What we see most of the time is that it's a little bit on the fungal side. Banana's a big tree grass. So it's a little bit on that side. Okay, what else? Petunias. Where do petunias occur out in the natural world? What kind of ecosystem? You might have to go look that one up on the internet. Google petunias and find out where they originally came from. Isn't that fun? Easy way to do it. Let's see. Marigolds. Where did they originally come from?
Probably in Nebraska.
Alpine meadows. Typically. Tomatoes, where did they originally come from?
Desert.
What part of the world? South America, right? So up in the Andes, we're looking at some dry grassland systems. So where are tomatoes in this? We're right here with the rest of the veggies. Anybody have a plant that they want to ask about?
Coconuts.
Coconuts. What kind of system are coconuts?
All grass.
Oh, grasses. So savanna conditions. They do fine with some grasses that are growing right up to the root system. But what does the coconut actually need? Because a lot of times when we look at savanna systems, we're looking at a situation where we got lots of grass out here, and then here's our tree, wherever it might happen to be. And so there's the trunk, lots of grass. But now let's start looking below ground. How far down do the roots of your grasses go? Let's give it 20 feet. How far down do the roots of your tree go? 100 feet? 150? Okay. depends. You need to know a little bit about that particular plant species. But now look at the fact that your tree is putting its roots down way deeper than the grass. So the grass may be slightly bacterial, or it might be equal fungi to bacteria, somewhere in that range. And the tree doesn't really kind of like that. So when it's competing with the grass, it tends to put those roots just straight down. And then once it's free of that competition, once it's out of that warfare, now the tree grows up underneath the roots of the grass. And this is extremely fungal. So just because it's growing with a lot of grass around it, we do have this division of your soil below ground where the grass and the trees are sharing space in a pretty friendly fashion. What happens to your tree if you put it into compacted soil? Your tree is never going to do well. It is always going to be competing with that grass, and now it becomes a question of who's going to win, the bacteria or the fungi. And you could have a tree that's going to always be sick and unhappy and unhealthy. So we want to make certain the roots of your tree can get down underneath the grasses that might be growing up to it. When that tree's really little, when that, you have a little whip that goes in there, and those roots aren't going down very far, you probably want to make sure that you have a space around that little tree where you're putting in good fungal foods. Fungal compost, fungal mulch, so that that root system is fungal, and then it grows down deep and under the grasses that it was competing with. And then we can have both things healthy.
So coconut, there's a palm, which is not a woody plant, really. It's really, it's from like more of a grass type thing.
And so I always have that with coconuts, with palms of various kinds. I had to actually go out into the real world and look at what's around the root systems of the palms. Very fungal. They are indeed very fungal. And then I have woody wood. You got some kind of a soft wood, but they really like it fungal. Question?
So ginger and ginseng are extremely fungal then?
Yeah, very fungal. Yeah, so those root systems, ginger, ginseng, where do they originally come from? Definitely forest systems. Pardon?
It's just interesting because they're heavily fertilized crops here. Yet the fertilizer would kill the fungus, right?
Yep. So a lot of times with that ginger, we have real problems with the diseases. And we're keeping them alive in our agricultural systems because we treat them like strawberries. We don't give them the conditions that they need to grow. A lot of times with ginger, you're happily nurtured it along for 2-3 years, and you're coming down the 4th year where you harvest it, and it all dies. So ginger in the Pacific Northwest is just a pain in the butt. How long do you grow ginger before you harvest it here?
1 year or longer.
A year or longer, so you probably don't have. So there's a difference between a cold, not a lot of sunlight environment, and here.
Yeah, but they keep going through fields.
Yeah.
So they'll put so much fertilizer on one field that they lose productivity and then they go to the next one, kind of like your South Africa example.
Because of the nematodes.
Yeah.
They get eating nematodes.
Slash and burn. And we need to do the things that make certain that those root-feeding nematodes are selected against. So let's get those other nematodes in the system. Let's get some mycorrhizal fungi. Those are the 2 things that really help select against the root feeders. How fast can you get rid of root-feeding nematodes? Let's see. Do I have an example here? I think we'll do that tomorrow. The difference between soil and dirt. We've been there, we've done that. Hopefully, all of you could explain this particular slide. Use it as something to introduce your talks or summarize your talks. Soil, organisms build structure. Dirt, no organisms, no structure. Nutrients are held. Water moving through soil comes out clean. It's drinkable. It is an ecosystem service of soil that it makes clean water. Dirt. Typically, you have a compaction layer, and your soil, your water is going to move sideways and cause erosion. Any water coming through dirt is going to be chock full of nutrients, all kinds of nasty stuff, and it's going to harm everything else downstream. Water is retained, moves slowly. Water moves rapidly through dirt. So the differences between soil and dirt. Gotta have life in there. So there is hope. We can very rapidly and very quickly recover all of the dirt that we've made and turn it back into soil. It's not going to cost billions or even millions. We can do it very rapidly. We just make good compost and get that compost into our soil in a liquid form or in a solid form. Get the whole food web back, and we can solve this problem. Ta-da!
Alright.
Thanks. Compost, extract, composting tomorrow.